switch over to Matt Cornice to address the mass marking program. We've had a lot of good data coming out of that program over the last several years on a few different species.
Well, thank you all for having me here today. I'm a biologist with the Mass Marking Program, work with the Fish and Wildlife Service out of Green Bay, Wisconsin, and I'm going to be sharing some results from our Mass Marking Program today. But before we get into that, for my outline here, I am also going to give a brief overview of some of the other programs we have within Fish and Wildlife on the Great Lakes out of our Green Bay office. And then I'll dig into the results we have for Chinook salmon, lake trout, and steelhead on Lake Michigan. So, a question I get a lot is, what does the U.S. Fish and Wildlife do? We are a federal agency, and our mission is to conserve, protect, and enhance fish, wildlife, plants, and their habitats for the benefit of the American people. The way we go about doing this is to work towards healthy fish and wildlife populations and habitats to provide quality hunting, fishing, and other outdoor wildlife-dependent recreational opportunities. And within the Great Lakes, at least at ARFWICO, our office's role is to assist states and tribes with management of aquatic resources and with invasive species prevention. So mass marking is one of the programs housed out of our office. Another one that many might be familiar with is our Aquatic Invasive Species Monitoring Branch for Lake Michigan. This group monitors for new invasives in Lake Michigan and its tributaries with a focus on silver bighead and grass carp detection using both eDNA and traditional nets and other gear to sample these fish. There's some other invasive species like rough and goby that they work on. They coordinate with the state agencies for rapid response plans should there be a detection. And within the Fish and Wildlife Service, there are comparable programs on each of the Great Lakes. We also have a native species branch in our office that focuses on monitoring for native lake trout abundance using multi-agency gillnet surveys, contributing to stock assessment models for both lake trout and lake whitefish, and then more recently contributing and participating in some of the GLADOS acoustic telemetry work in Lake Michigan to look at fish movements and habitat use, and then also mapping of habitat using a multi-beam sonar to get kind of updated bathymetry in various parts of these lakes. Third, we have a partnerships and habitat branch that works primarily in the tributaries. They focus on stream connectivity to help with fish migrations, basically looking at culvert and dam modifications or removals. They also are engaged in land grants to protect habitats as well as easements for access, involved in some brook trout restoration and some native mussel conservation, as well as assessing and prioritizing various risks in these habitats. And then kind of fourth, we have a lake sturgeon restoration program that participates in streamside rearing and release, as well as research on adult sturgeon, and helping to operate the fish lift on the Menominee River to move adults upstream of the dam to where there's better spawning habitat. So that's kind of a whirlwind overview of the things the Fish and Wildlife Service at our office is working on. The last branch is the focus of the talk, the Great Lakes Fish Tagging and Recovery Branch, which is more commonly referred to as mass marking. So mass marking program, this is a collaboration among federal, state, and tribal agencies. It's coordinated by the Fish and Wildlife Service here in Green Bay. We started in 2010 to address questions on the survival and contribution of hatchery fish, natural reproduction, movement, growth, and other measures for salmon and trout. States have management authority, so the Fish and Wildlife Service helps to coordinate this research across state jurisdictions, but the state DNRs are pulling the levers on regulations and stocking numbers. Our program is funded by the Great Lakes Restoration Initiative, which comes to us via the EPA. And thanks to your support and from other anglers, we have 12 years of data on over 172,000 fish from open water angling, which gives us a very powerful data set to get at some of these questions that are of interest to both managers and stakeholders. Our program has four major components, which briefly include the tagging and marking of hatchery fish using our AutoFish trailers as seen here. Data collection and recovery from the sport fishery, which is where you may interact with our biotechnicians in the field, hoping to collect data from your catch. Back in the lab during the fall and winter, we work on extracting tags from these fish, as well as aging wild fish and processing of other lab samples that we may be collecting for partners like the Brian Roth-led diet study you just learned about. And lastly, we do data analysis and support for managers. Talking a little bit about tagging, this figure shows the number of fish marked in millions per year. And you can see since the inception of the program, we've primarily worked on lake trout, which you see in green, Chinook salmon in blue, and steelhead in pink. And on the whole, we are marking about 10 million fish every year at state and federal hatcheries, primarily from these three main species. Now, starting next year, actually starting this year, we have stopped tagging steelhead, and at the request of the states, have moved the funds used for steelhead to start tagging coho salmon. So we actually have one more coho salmon tagging trip at Platte River Hatchery coming up in June. Otherwise, we have had these fish tagged and marked at all the other state hatcheries in Michigan, Illinois, Wisconsin, and Indiana. A question I often get about the marking trailers is what do they look like on the inside? And so I do have a couple of brief videos here to show kind of this tagging part of the operation. On this first video here, and I might have to tweak my volume so it's not too loud. Let's see here. So you'll see fish coming up into this tube, up to this sorting pen up here. Basically, these fish are swimming upstream. We're utilizing that to have them kind of direct where they need to go. This is a length sorter, so these fish are flying across this optical device that takes rapid length measurements. That was the red light you see there. All that clicking you hear is the fish being sorted into different lines. These are each calibrated to a specific size range of fish. My kids love these videos because it looks like the fish are going down these water slides. They go into these holding pens where, again, there's water coming upstream into those pens that encourages the fish to swim into the tagging unit, which we're seeing here. This brown device comes down and clips off an adipose fin, while at the same time they receive a coated wire tag in their snout. These machines come with a lot of quality control devices. So there's a before and after image taken to ensure that the clip has been removed. And there's also a tag detector in the tube to make sure a tag is present. If either of those things are not present, it gets kicked out to a reject bucket where we evaluate those fish by hand and give them the clip or tag that may have been missed by the machine. On the whole, we tag, our success rates are about 99.5% for fin clips and about 96% for tags coming out of these trailers. This other video shows you a little bit about the outside operations. Turn that volume down a little bit. So this here, I believe this is at the Iron River National Fish Hatchery up in Iron River, Wisconsin. You can see the raceways here full of fish. What you're seeing here is a very large water pump. This is pumping all of the water into the trailers. The main thing I want to show you is this video. If we continue and skip a little bit ahead, walking down the side, you can see this pipe leading to an empty raceway any minute now. You can see all the fish here. That's always a cool sight. But this pipe is going to an empty raceway. It's filling up with the fish that we have tagged from our trailer. And this little hatch in the back will show you the fish coming out of the trailer. This is where we sample to make sure we have successful fin clip and tagging rate. On the whole, these trailers process about 8,000 fish an hour, which is about 2 to 3 fish every second. And that's how we're able to process so many millions of fish every year. The second main component of this program is the recovery of tags and data from the field, primarily from anglers as these fish are returning. The main thing I want to show you here is this map. All the red dots on this map are the ports that we survey annually in Lake Michigan and in the U.S. waters of Lake Huron. So collectively, we get a lot of biodata. This has been one of the most abundant sources of data for many of these fish. On average, we're getting between 10 and 15,000 observations a year. And because we put our hands on so many fish, it lets us help out with things like diet studies and other studies. Get the tags back out, that's a manual process. You can see a picture here of one of our techs pulling a tag out of a snout. So far, we've recovered a little over 157,000 tags from our sport fish survey, as well as from agency gillnet surveys, commercial fishermen, and voluntary angler returns, as well as WEIR surveys conducted by the states. On the right here, you see a kind of a zoomed in picture of one of these tags. You can see there's a code that's etched on it. Each tag is a very small, filament section of wire that's maybe a 64th of an inch long. And the code corresponds to a batch of fish. So this code 640185 is telling us that this particular fish was one of about 200,000 lake trout that were stocked on the Southern Refuge in 2010. with, I believe, a Seneca Lake genetic strain. So that's kind of information we get out of these tags. We also look at scales and otoliths to age these fish. I have a very nice picture of an otolith here that kind of shows you how this works. You can look and count rings on a tree. Now, admittedly, this otolith picture was picked because of how clean it is. Sometimes these estimates are a little more challenging to make, but nonetheless, we're able to do this to get estimates on these wild fish, which is an important component of a lot of the stock assessment models used around the lake. And then lastly, for data analysis and support for managers, I just briefly want to touch on how these data have helped answer these questions about wild recruitment, movement, survival, and growth for Chinook salmon, lake trout, and steelhead. Data that we produce are regularly used in the models that inform state management and policy decisions on stocking and harvest regulations. And in addition, I already mentioned the diet study, but we are able to collaborate on a lot of different things, looking at sea lamprey wounding on different fish. You heard about the salmon and trout diet study earlier. We've also engaged in a couple of studies where we're able to look at the natal origin of wild steelhead, Chinook salmon, and coho, basically looking for which tributaries are producing those wild fish, using a really cool method that basically matches the chemistry of these otoliths or ear bones in the fish with the chemistry of their the streams in which they hatched from. We've looked at what genetics are contributing to wild lake trout production in the lake, and most recently we've had some contribution to the detection of PFAS or PFAS biocontamination in fish in the Great Lakes. So I don't have a ton of time to go into all of those things, but just wanted to give you a broad scope of several of the things that we do. Moving right along, for our first focal species, I want to talk about Chinook salmon. So we've been working on Chinook salmon since the 2011 year class. One of the things that we discovered first from all these tags is that Chinook salmon are moving all over the lake for most of the year. Now, if you think about the Chinook salmon, they're native to the Pacific Ocean. The Great Lakes, while big, are smaller than that, so it's not much of a surprise that they move as much as they do. But nonetheless, it doesn't really seem to matter where a fish is stocked. It can be recovered anywhere. So what you're looking at here are two different maps. This map on the left, the Gold Star in Manitowoc, shows you where these fish were landed, and the red circles correspond to the number of fish stocked in each of these areas of the lake that were landed by anglers in Manitowoc, Wisconsin. So you can see that these Chinook that were caught in Manitowoc were stocked all over Lake Michigan as well as in northern Lake Huron. And we see a very similar pattern if we move across to Frankfort, Michigan. That map on the right is showing landings in Frankfort. Again, those circles are corresponding to the size of number of fish from each stocking location. Another way to look at this kind of a little more locally is I put together some pie charts for Chinook salmon that have been landed in Muskegon and Grand Haven. This is also known as the MM7 unit in Lake Michigan. So from 2014 to 2019, which is the period of time when we were tagging all Chinook, we observed 889 tagged Chinook salmon landed in Muskegon and Grand Haven, and I've got their stocking locations here for you. I should mention we started to tag all Chinook salmon again this year, so we're reevaluating fresher year classes and updating the data here. But nonetheless, so if we look at April through July, this pie chart is showing where these fish were stocked. Again, these were all stocked fish that were caught with tags in either Muskegon or Grand Haven. The green bar are fish that were stocked in Illinois. Purple bar are fish stocked in Indiana. That red bar is actually Chinook salmon stocked in Lake Huron that moved over to Lake Michigan. Gray is fish stocked in Wisconsin. This light blue bar are the locally stocked fish. Those are the Chinook salmon that were stocked in Muskegon or Grand Haven, and they comprise about 10% of the landings. Dark blue are fish that were stocked elsewhere in Michigan. The one thing we learned, though, is that these fish do appear to have natal homing, even though they are only in their stocking streams for a couple of weeks after they are stocked. They're there right at that critical smolt period when these fish pick up on the whatever they're picking up on in the environment to cue in on where they're supposed to return to. So as we transition into August, we see a very similar pattern, but once we get to September and October, at least for these stocked fish, you can see the majority of those landings were actually stocked locally in Muskegon and Grand Haven. I mentioned a little bit about the movement. You saw a lot of Lake Huron fish being recovered there in Muskegon and Grand Haven. One of the things we learned is that after alewife collapsed in Lake Huron in the early 2000s, we started to see that Chinook salmon were moving from Lake Huron into Lake Michigan during the feeding season. So this map on the left shows Chinook salmon that were stocked in northern Lake Huron, MH1, which I'm trying to circle with my mouse here. And we can see these circles are proportional in size to their recoveries. You can see most of these Lake Huron stocked fish were captured in Lake Michigan, with most of them actually in northern Lake Michigan. Now, we think the reason for this is that we do have more alewife in Lake Michigan at this point, and Chinook like to feed on alewife. But it's also possible that we're just observing the fish that were able to survive, and those just happen to be the ones that are moving into Lake Michigan. Either way, there's not much of a reciprocal movement. I think over the course of the years, we've only captured like 5-ish fish that were stocked in Lake Michigan that somehow got lost in a Lake Huron. So it's kind of one directional. You can see that the data point I have here is about 94% of those Chinook salmon stocked in Lake Huron were recovered in Lake Michigan during feeding. However, if you look at this map at the right, these are these same fish in August and September. You can see that biggest dot is here in northern Lake Huron. And so that tells us that these fish are also returning to Lake Huron during the spawning run. One thing we estimate every year is the wild recruitment of Chinook salmon. We do this by looking at the ratio of hatchery to wild fish. Hatchery fish is going to have that adipose fin removed, as you see in the top photo. A wild fish should have that intact, as we see in the bottom photo. And I want to say again that about 99.5% of hatchery fish are successfully fin clipped. So we're very confident that most of these fish with a fin are wild. And that's also been confirmed by both us and by independent anglers using tag detectors on unclipped fish. So it might surprise you to learn that we typically see anywhere between 60 and 70% of the Chinook salmon in Lake Michigan as being of wild origin. We had one exception to that in 2013, and we'll see that in the next figure. But by and large, we're at about 2/3 of those fish being wild at the lake-wide scale. So the way that we can estimate wild production is we know how many fish were stocked, and we have from our observations the percentage of wild fish, so we can use a ratio to figure out how many wild fish must have been present to have produced the percentage of wild that we observed. And so in this figure, we have stocked smolts in orange. That's a known number. Blue smolts, the blue bars are the wild smolts, which we estimate from that procedure. And you're looking at the number of the millions of smolts on the y-axis per year class on the x-axis. Two things I want to note real quick. We are asked a lot about, well, 2020. So the 2020 year class did not get tagged, did not get clipped. And so the value for 2020, you can see the shading is a little bit different. That's actually just an average of the 2018, 19, and 21 year classes. It's just a kind of a filler to try to estimate what recruitment might have looked like there, because we just don't have the data. The other thing to note here is the 2023 year class. You may have caught on the prior slide, we conduct our formal estimate when these fish are returning at age 2. That's because that's kind of the most abundant size class that we see. 2023 these are 1-year-olds. These are from last year's 2024 data. So this number is estimated from age 1 fish, but will be updated with this coming year's recoveries. So if I take the stocked smolts off of this figure, well, this is actually, we're showing about 7 million wild fish from the 2022 year class and about 9 million from the 2023 year class. But what I want to focus on now is kind of the variability in wild recruitment for Chinook salmon. And the easiest way to do that is to take those stocked smolts off the figure. And when I do that, what I see are 3 kind of major patterns here. Early on, we had 7 year classes where we had wild production ranging from 6 to 8 million wild smolts a year. And then we had what happened in 2013. During the fall, there was a severe drought in about half the lakes. There were record low returns at many of the weirs. And as a result of that, spawning activity was more limited for that year class. And that was combined with a very harsh winter where there was kind of a polar vortex hanging out here. I remember it well because I moved back to the Midwest from Maryland in the spring of 2014, and it was like May 15th and 30 degrees with no leaves on the trees out. So that could have delayed the ability of these wild fish to get their food during a critical point. For whatever reason, that was a historically weak year class. And then in the subsequent year classes, we likely had fewer adult fish returning to spawn because of that weak 2013 year class. So during that period of time, we had about 3 to 5 million wild fish. Um, but over time, we've kind of returned in these last several year classes to about the same range we had seen in the first part of the time series. And so this is part of why we do this every year. It's because there is a lot of variability in wild recruitment, even though it looks like there are some patterns we can tease out. So I mentioned earlier on these fish that were recovered in Muskegon Grand Haven, and I was only looking at stocked fish, but I want to also note that we recovered a lot of wild fish in these areas. And just to reinforce what we've talked about already, if we add the wild fish to these pie charts, again, these are Chinook salmon landed in Muskegon and Grand Haven, and we can see that the amount of stocked fish is only about a third of those fish being landed. In each of these time frames, we see a substantial number, about 2/3 of the recoveries being of wild origin. And what's interesting is if we move farther north into MM6, which has Ludington and Onekama in it, we actually see more wild fish returning in the fall. And you can see that yellow bar grows and grows as we proceed from April through September and October. And this is kind of the first signal to us that there's a lot of wild production for Chinook salmon going on in northern Michigan. So when we look at these fish in Wisconsin in a general sense, this is the percentage of fish captured on the y-axis. And the key lines here, we have red are fish that were stocked nearby, generally less than 10%, but that rises into the spawning season, like we saw for Grand Haven and Muskegon. And then in Wisconsin, we see that these wild fish are about 2/3, about 65%, except then we get to the spawning season and we see them vanish. And what's actually happening is they appear to be moving into Michigan waters. So in this figure, we have the wild Chinook salmon Wisconsin line, and then I've added a wild Chinook salmon Michigan line. You can see the numbers are very similar through about early August, and then we start getting into this transition in the spawning season, and those wild fish appear to be moving back to Michigan. This is something we were able to confirm by looking at the catch per effort rates in the fall. This heat map is showing the average wild catch per effort in fall in Lake Michigan. The warmer the color, the more wild catch we see in the fall. And you can kind of see Northeast Michigan being a focus for where these wild fish are coming from. With these tags, we're also able to try to estimate survival rates based on the number of returns per number of fish stocked. The main thing we found here is that there's high relative survival for fish stocked in Wisconsin, which we think is due to colder water temperatures and perhaps better forage availability at different life stages. We see really low survival, these pale yellow colors in Green Bay. There's a lot of predators there and it's warmer water. And also in MM6, which in talking with a lot of the managers over in Michigan, the egg take facility there on the Little Manistee River is several miles upstream of the lake. And so the fish that are stocked there have to go through a bit of stream as well as that drowned river mouth, Manistee Lake, where they may be subject to predation. And so that's, as well as the fact that there's a lot of wild fish there, potentially competing with these stocked fish, might be the reason we see lower survival of fish stocked in MM6. Another thing we've noticed in this study is that there was a decrease in survival from the 2011 through 2016 year classes. And this is a time series we're going to hope to pick back up now that we're tagging Chinook again with the 2025 year class. So this information is always provided to the states to help assess the stocking programs and to provide information used for various decisions on stocking. With that, I want to move on to data we've collected on lake trout. So we look at the relative abundance or catch per sampling day as a way of looking at abundance of fish. For lake trout, we kind of looked at 3 large regions just to kind of condense things a little bit. We have Southern Lake Michigan in dark blue, Northern Lake Michigan in light blue, and Lake Huron in orange. What you're going to see here, we only have values from districts where we felt we had enough fish, at least 20 lake trout observed to provide a data point we have some confidence in. And so what you're seeing in this figure is the abundance of relative abundance of wild lake trout in our sport fishery observations over time. You can see it's increased in all three of these areas, but it's increased most substantially in Lake Huron, with a modest increase in southern Lake Michigan, and then more recently, after seeing pretty much bupkis through 2018, a bit of a small increase in northern Lake Michigan as well. If we add on the hatchery fish, so I'm going to go through each of these three systems using kind of the same format. Again, we have the relative abundance on the y-axis. In each of these figures, we have the total lake trout in black, wild in gray, and the hatchery fish in orange. These are the recoveries in Lake Huron. And so here we can see that increase in wild lake trout in gray, while catch of hatchery lake trout's been more or less stable, in part because stocking levels were more or less stable during the time period leading up to this. There was a decrease from 2012 to 2013, which I think was related to some stocking cuts. But our total lake trout catch is really being affected by the wild lake trout in Lake Huron at this point. If we move west into northern Lake Michigan, this is the unit where we have sort of the least amount of data, and that's contributing a little bit to how these patterns bounce around, because in some years we couldn't include some of the districts. But the main thing to take home here is that the total lake trout catch has closely mirrored the hatchery lake trout catch for this entire time period. And the wild fish, while we are happy to see some improvement there, have been very slow to take hold in northern Lake Michigan, in part because of harvest and in part because of sea lamprey abundance. And then if we move into southern Lake Michigan, we get kind of a very interesting pattern where we have hatchery lake trout that were relatively high, but then declined. And that decline is related to stocking cuts we saw in southern Lake Michigan from, I want to say, 2004 through about 2008. And then along that same time frame, we had an increase in the abundance of wild fish, which is a good thing. But we can see that the total lake trout was mirroring patterns in the hatchery fish, but now is mirroring more patterns in the wild fish here in southern Lake Michigan. I think I left this figure in. Yeah, so one thing I want to talk about, we used to talk about the percentage of wild fish as a measure of how wild production was going for lake trout. But we had to move away from those metrics in large part because of these data. A percentage is going to be based on both the hatchery and the wild abundance patterns. And so this figure, you see the southern Lake Michigan hatchery and wild trout, and overlaid on that is the percentage of wild fish. And as the hatchery lake trout have declined, you can see that that percent wild is looking less and less like the actual trend line for the abundance of wild fish. It's producing an overestimate, even though we have more than 50% of those trout in southern Lake Michigan are wild at this point. We are seeing a decline in the total catch because of that hatchery lake trout trend. So I just want to highlight why we're not talking about percent wild anymore. You can see here how it could potentially be misleading for how wild lake trout are doing in the lake. Some other things we've learned about lake trout. Unlike Chinook salmon, lake trout don't move as much. I mean, they are fairly mobile, but their dispersal range is limited to about 100 kilometers or 62 miles. And so, for example, these are lake trout that were stocked in the unit WIIM, which is shown here in pink. It's called that because it has Wisconsin and Indiana and Illinois and a Michigan unit. So scientists, being the creative folks that we are, just called it WIIM. These black circles are proportional in catch to those fish stocked there. And you can see that most of the lake trout that were stocked in WIIM were caught there with less and less fish the more northward you go, which kind of shows that limited dispersal for lake trout. Fish that are stocked offshore do seem to be a little more mobile because their movements near shore are a little more sporadic, whether they go east or west off of refuge. So from this, we've learned that offshore refuges are source populations for nearshore fisheries. This map is showing lake trout stocked in the northern refuge as blue circles and lake trout stocked in the southern refuge as red circles. And you can see that between these two refuges, they're providing lake trout to pretty much all nearshore areas of Lake Michigan. But the largest circles are still within those respective refuges. So we do have evidence here that a portion of those lake trout that are stocking refuges are remaining there and therefore receiving some protection from harvest. As a result of that protection, and as a result of the fact that these refuges are on really good lake trout habitat, we do see higher relative survival for lake trout stocked offshore. So offshore stocking sites are color-coded blue. Near shore are warmer colors, and you can see the corresponding units on this map at the right. In particular, fish stocked at Julian's Reef in Illinois and the Southern Refuge have the highest survival rates for lake trout stocked in Lake Michigan. And those stocked near shore tend to be a little bit less in terms of their survival, especially for those that have been historically stocked in the Wisconsin shoreline for whatever reason. And then I'd like to close with the time I have left talking about steelhead, which is the most recent fish that we've tagged, trying to learn similar measures for steelhead as we have seen here for Chinook salmon and lake trout. So we are able to look at the wild production of steelhead in Lake Michigan. From our observations from year to year, the overall percent wild has ranged from 32 to 50%. And what we're happy to see is there actually was a completely independent study using the otolith microchemistry that we talked about a little bit earlier that classified about 40% of their observations as wild fish, which is like dead center of our range. So it always instills confidence to get kind of similar numbers from two entirely different approaches. And so we're pretty confident in how we're estimating wild production of steelhead. Now, here we have the same figure that we had for Chinook. The orange bars are the known number of stocked smolts. The blue bars are our estimate of wild smolts. 2020 was an interesting year in that there was no egg take in Michigan. I did talk to Ben Tershak about that. He confirmed that the stocking database was accurate. Because there was no egg take in Michigan for that year class, there wasn't a steelhead stocked for that year class in Michigan, and that sort of makes 2020 year class an anomaly. So I have it as a different color pattern. But by and large, we see a little bit of variability so far. The first 3 year classes were around 1.5 to 2 million wild fish, whereas the more recent 2 year classes were about 1 million or less. So it remains to be seen how this trend line will progress, but this is something we are looking at every year with our annual collections. In terms of movement, we've found that steelhead are much more similar to Chinook salmon than lake trout, which makes sense to us because steelhead are also a Pacific salmon. They're actually in the same genus as Chinook salmon, and they are mixed during non-spawning season. So the example I have for this, this figure shows steelhead stocked in Indiana, which you can see highlighted in yellow, and the circles are corresponding to the various catch rates. So those Indiana stocked fish are being caught by anglers throughout the lake. In terms of what's going on we also saw a lot less movement from Lake Huron to Michigan compared to Chinook, though. Only about a quarter of the steelhead from Lake Huron are observed in Lake Michigan, and we think this is because of steelhead having a much greater variability in their diets with some of the things Brian talked about with consumption of invertebrates and the like. If we want to see what happens with the steelhead that are stocked in Michigan waters, I have those maps here. And the take home here is that those fish are being caught throughout the lake as well. So the left map shows steelhead stocked in northern Lake Michigan. The middle map are fish stocked in kind of center Michigan, kind of the Ludington, Manistee, Grand Haven area. And then the right map shows fish stocked in southern Michigan waters. And in all three cases, you can see the catch of those fish represented by the circles is occurring lake-wide. So, kind of a quick way to summarize this is asking who's catching whose steelhead. And the answer is everybody's catching everybody's steelhead. All the states are dependent on all the other states for their catch. You can see these pie charts are steelhead caught in Illinois, caught in Indiana, caught in Lake Michigan waters of Michigan, and caught in Wisconsin. And with the exception of Indiana, where we see a higher representation of Indiana stocked fish, these pie charts look very similar. And when we are asked, well, why do we see these patterns? It's fairly simple. This is a pie chart on the right showing the total stocking in each state. So we can see that they're very similar here. The stocking numbers are corresponding to the catch, with the exception that the Indiana fish are maybe a little overrepresented in each state, which is probably because of their survival rates, which we're going to get to. But otherwise, by and large, we see that steelhead are being caught everywhere and in about the proportions you'd expect based on stocking numbers. So I did kind of mention survival. Methodologically, we had to do what's called a standardization because we looked at survival of fishes age 2 through 4, but because of the COVID 2020 season, we didn't have the same aged fish for every year class. So without getting too technical, the bottom line is with these values, the value of 0 is going to be the average for that year class, with positive and negative values corresponding to above average or below average survival, with a single unit like +1 and -1 are equivalent to the standard deviation, if you guys are stat nuts like myself. So this basically removes some of the bias that we would have expected from using different ages in the year classes. One of the first things we learned is that yearlings are surviving at a much higher rate than fingerlings. So this figure is showing that relative survival. Again, 0 is average. You can see fingerlings are by and large having below average survival. Yearlings are by and large having above average survival. And it turns out it's about 2.5 times greater for yearlings than fingerlings. So another way of putting that, fingerlings represent about 23% of the steelhead stocked in Lake Michigan, but only about 7% of the sport harvest. And nonetheless, a lot of times these fingerlings are stocked because the hatcheries need to make room for the next species of fish. And so that's why some of these are stocked as fingerlings instead of yearlings. But because of this pattern, for the rest of our survival patterns, I'm looking exclusively at yearling fish. So this figure, which I'm not going to spend a ton of time on, is actually showing the standardized relative survival rates for all the stocking units in Lake Michigan. And I just want to show you how I came up with the color codes you're going to see on the next map. We have 3 units in blue that had substantially higher survival and actually statistically significant higher survival compared to the red box. We have a yellow box of fish that were more or less average survival rates or slightly above average. And then this red box are units where we had really poor survival. The easiest way to look at these data are on a map. And so this map on the left is the exact same data, but expressed visually. And so from this, we can see we actually have the highest survival of steelhead in Lake Michigan are stocked in Indiana and in southern Michigan waters. And for whatever reason, Wisconsin, they do stock some of these steelhead throughout the state in very small tributaries. They all share the same tag code, and that particular tag code survived very well as well. But we don't know where exactly those fish were stocked, and that's why that box is over here, kind of in the middle of Wisconsin. In terms of our low survival, like Chinook salmon, we have very low survival for fish stocked in Green Bay. We think the same factors for Chinook, the predation levels and the warm water temperatures are what's explaining this pattern. For WM4, the other red unit here, I am kind of at a loss for why we see such low survival there. It could be a hatchery effect. It could be any number of things, and we're still trying to figure out what's going on with that. The map on the right is just showing, again, with circle sizes where all these fish are stocked. This map was taken directly from the Great Lakes Fish Stocking Database, which you can access online and play around to look at these stocking numbers for all the fish stocked in the Great Lakes. So if you're interested in that, Google Great Lakes Fish Stocking Database and it'll take you to that website. We have not seen any significant differences among genetic strain. But with comparing Skamania and Michigan winter strain, we're actually getting kind of close in some of the units. So this map is showing survival of Skamania strain in blue and Michigan winter in orange when they're stocked in the same unit. So we're controlling for stocking location a little bit. And in 2 of these 3 locations, we did get a higher return rate from Skamania. Still not statistically significant, but with additional years of data, if they trend the same way, it might push that into a significant trend. But as for now, there really wasn't anything to speak of in terms of genetic strain differences. This was the strongest difference we see when controlling for stocking location. In Wisconsin, there are 4 units where they stock both Chambers Creek and Ganaraska strains with very similar survival rates. And in Wisconsin, there are 2 units where they actually stock 4 different strains. Arlee, Chambers Creek, Ganaraska, and Skamania. And again, we see very similar survival for those strains when controlling for location. So to conclude for steelhead, all states are depending on one another's steelhead. We see pretty poor survival in Green Bay and somewhat poor survival in WM4, which is where Algoma and Manitowoc are in Wisconsin. Good survival for Indiana, MM7, which is that Muskegon, Grand Haven area, and the Wisconsin small tributaries. And no significant differences detected among strain. So with that, I think I only have a few minutes left. I'd be happy to take questions. I'm going to kind of skip through the bonus material because I don't think we have time for it. But that was the core talk. And thank you very much for your attention. My contact information's up there if anyone wants to reach out. Otherwise, Dan, if I have a few minutes, I'd be happy to take questions.
Yeah, thanks, Matt. I have not seen questions come in yet on the Q&A or the chat specific to you, but I did want to ask if you've looked at steelhead survival through the years and if there's been any increase in the survival of stock steelhead since this began.
Yeah, so that's a very good question. We haven't looked at that as of yet, in part because we're just getting through we had that COVID year where we didn't have tag steelhead right in the middle of the original study design period. Um, we haven't seen a lot of year class differences in survival, except that I know early on we didn't see very many Wisconsin fish to speak of. And that's in part because they stock Anarascas, which tend to be a little bit smaller at age 2. Once we started getting older age data, we started seeing more of those Wisconsin fish. But in terms of a time series of survival, that's something that we should look at and have for you next time around.
Thanks. And not seeing any other questions, but one other thing that we might want to address is the future of the program. And I know there's been a bill introduced to fund this. I think one of the questions I've heard in the past is why not mark all the fish every year? And I think that probably comes down to budget, right?
Yeah, it absolutely comes down to budget. So, I mean, the tags run about 10 cents a tag, but we're doing 10 million fish. So, our annual budget is enough to tag at this point all of the lake trout plus 2 other species. And so this year we're tagging lake trout, Chinook salmon, and coho salmon for Lakes Michigan and Huron. To tag all of the species would require, more funds. And you mentioned that bill that's been introduced to provide base funding. I mean, that's a positive development for the program to see that support in Congress. But the future of the program does depend on on budgets and staffing levels and decisions made way above my pay grade. So
Yeah, thanks. Appreciate that. And I think we're just about at 105 here. So thanks, Matt, for giving us an overview of the great work being done by the Mass Marking Program. Thanks, everyone, for attending.